welcome to today's video lecture. Today's video lecture, I'll be solving previous year's NEET questions and discussing the answers. And it's for the chapter Biotechnology, Principles and Processes. And we are in part 4 of the series. So if you haven't checked out part 1 to 3, please do so. The links will be there in the description box. Alright, so let's begin. Okay, so question number 1. Recombinant DNA is achieved by cleaving the pro-DNAs by, this appeared in the year 1998, A. ligase, B. restriction endonuclease, C. primase, and D. exonuclease. So recombinant DNA is achieved by cleaving the pro-DNAs by restriction endonuclease. Okay, so the answer is B. So I've already previously explained how our DNA is formed, right? So if you have a pro-DNA and you want to take a desired fragment from here, let's say your fragment is over here. Okay, so a restriction endonuclease might cut over here and another over here. Okay, and then you can get that particular fragment and then put it into the plasmid over here. Okay, let's say if just change this color into a different one. Okay, so you have this over here. Okay, so this is your desired fragment, DNA fragment, which contains a gene that encodes for a particular protein that it will express, and this is the plasmid, right? So what you get here is a rDNA. Okay, so you need restriction endonuclease for that. All right, pretty simple, right? Let's move on. Question number 2. Two bacteria found to be very useful in genetic engineering experiments are, and this appeared in 1998. Let me just change this. A. Nitrobacter and Azotobacter, B. Rhizobium and Diplococcus, C. Nitrosomonas and Klebsiella, D. Escherichia and Escherichia and Agrobacterium. Okay, so that are useful in genetic engineering are D. Okay, so the answer is D, and we've already discussed this also. Yeah, so E. coli, also known as E. coli, used as a host, okay, competent host, in which the rDNA is transformed, into which the protein is expressed. Also, E. coli, from E. coli, many restriction endonuclease have also been found. So that's another advantage of using E. coli. There are many endonuclease, EcoR1, EcoR5, so on, okay, that have been found in E. coli. So that's another usage of E. coli in genetic engineering. And Agrobacterium also is a natural genetic engineer for plants. Okay, that also we discussed multiple times, right? Which infects dicot plants and contains a TI plasmid, tumor inducing plasmid. Okay, so if that plasmid can naturally absorb itself into that genotype of the plant and then express that particular protein, if it is been modified to form an rDNA. So normally TI plasmid would cause tumor in the plant, but we don't want that part, right? So we remove the tumor infecting DNA and put in our desired gene into it and then put it into the plant so that it can express that particular protein in that. Okay, so it is now used a vector to naturally genetic engineer plants. All right, so yeah, according to the options that are given below, it's D. Okay, let's move on then. Question number 3. Restriction endonucleases are, this appeared in 98, A. used for in vitro DNA synthesis, B. used in genetic engineering, C. synthesized by bacteria, D. present in mammalian cells for degradation of DNA. Now, there are two possible answers to this question. Okay, I would say yes, of course, they're used in genetic engineering. Restriction endonucleases are also synthesized by bacteria. Okay, and it was one of the ways by which it was discovered. Restriction endonuclease was discovered, and they inhibit viral replication. So possible answers are B and C, although many cases I've seen used in genetic engineering. If you have to look for the most appropriate answer, then or the safest answer, yeah, restriction endonuclease are used in genetic engineering. Okay, so ideally, yeah, B would be the right answer, but also there is a C option for that. So it was first discovered in E. coli, restricting the replication of bacteriophage by cutting the viral DNA. Okay, and they are bacterial enzymes that cut or split the DNA at specific sites. That's what it was discovered, and therefore they are now used in genetic engineering because of this particular property. Okay, so they are synthesized by the bacteria, but then eventually they are used for genetic engineering. So I won't say that both the answers are wrong, but if you have to really make a decision between B and C, then just choose B for the most safest option. Okay, and the most obvious option, yes, they're used in genetic engineering. But I really wanted to pinpoint that yeah, they are synthesized by the bacteria. So of course they restrict viral replication, known as restriction enzymes or restriction endonuclease. Okay, and yeah, like we all know, they are also used in genetic engineering. Okay, so question number 4. The restriction enzymes are used in genetic engineering because this appear in 95. A, they can cut DNA at specific base sequence. B, they are nucleases that cut DNA at variable sites. C, they can degrade harmful proteins. D, they can join different DNA fragments. Okay, so again, we've discussed this multiple times, right? What is the function of restriction enzyme? They can cut DNA at specific base sequence. The clue here is specific base sequence. Okay, so the answer is A. Then I'll not go into much detail. Sorry. Sometimes this thing goes crazy. Okay, so I'll not go into much detail. They cut at specific positions within the DNA molecule. All right, let's move on. Question number 5. Match the organism with its use in biotechnology. This appeared in NEET 2020, pretty recent. A, Bacillus thuringiensis is proteins. Thermus aquaticus is a DNA that polymerase, stack polymerase. Agrobacterium tumefaciens is cloning vector. Salmonella typhimurium is construction of our DNA molecule. All right, so it's A4. Yeah, it's B. A4, B3, C1 and D2. Okay, so the answer is B. Yeah, answer is B. All right, so Bacillus thuringiensis, okay, is a bacteria that contains these 3 genes, okay, and they are called as cry genes, okay, CRY, cry genes, okay, and they encode for this toxin, BT toxin. Now, this toxin is very, very important because it behaves like an insecticide. So if the insect eats that particular toxin, it dies. Okay, so that's why it behaves like an insecticide. You don't need to use insecticides for that. You can incorporate this particular gene into that plant, and that is that behaves like an insecticide. So it's safe from the insect. The plant is safe from the insect. Okay, so they are crop and insect specific genes that code for the toxin. Okay, so there are some genes that are useful for cotton, maize, and so on, but I'm not going to those details now. But just understand that Bacillus thuringiensis encodes for that BT toxin, and they contain these 3 genes. We'll study this more in detail when we look at the applications of biotechnology, but for now just understand this. They have incorporated this particular question into this, but anyway. Yep, so, and Taq polymerase is a DNA polymerase enzyme and isolated from Thermus aquaticus, okay, which is used as DNA polymerase in PCR. So it's a heat stable enzyme. Okay, a very important property of this particular enzyme, it is heat stable. Okay, and Agrobacterium tumefaciens is a vector, so we've already studied that before. And of course, the construction of the first rDNA emerged from the possibility of linking a gene coding antibiotic resistant with a native plasmid, okay, of Salmonella typhimurium. Okay, so that was the first rDNA molecule constructed from salmonella. Okay, so a plasmid was removed from salmonella and it was linked with, say, it was linked with an antibiotic resistant gene. Okay, so this is your antibiotic resistant gene and this is the plasmid from salmonella. All right, so that was the first rDNA molecule. This particular information is there in NCRT, so you can't miss it. All right, and this appeared in, of course, NEET 2020, so it's pretty recent question. All right, I think we've answered the question. Yeah, we can move on. All right, question number 6. DNA precipitation out of a mixture of biomolecules and can be achieved by treatment with, this appeared in NEET 2000 and, sorry about that, NEET 2019. Okay, so this A chilled chloroform, B isopropanol, C chilled ethanol, D methanol at room temperature. So DNA precipitation out of a mixture of biomolecules can be achieved with the treatment of chilled ethanol. So the moment you add chilled ethanol to the mixture, you will be able to isolate DNA. Okay, it precipitates out. I think there's a photograph given in NCRT where they have shown how, you know, they spool the DNA out. It's like a whitish structure, so you can check that out. And of course, this appeared in NEET 2019. So the information is there in NCRT. So the, of course, the answer is C. Yeah, addition of chilled ethanol results in precipitation of DNA. It is a fact. Please learn it. All right. Okay, let's move on then. Question number 7. Which one of the following equipments is essentially required for growing microbes on a large scale for industrial production of enzymes? This appeared in NEET 2019. A. Bioreactor, B. BOD incubator, C. Sludge digester, D. Industrial oven. Okay, so yeah, for producing enzymes at a large scale, industrial level scale, then you need a bioreactor. Okay, so the answer is A. So this is what a typical bioreactor looks like. Okay, it's they used to convert raw materials into biologically converted specific products. So now till now we have studied how an rDNA is produced. Okay, how it is formed, and once you have that rDNA, you want it to express the protein, right? So they obviously, you need like the culture medium, you need pH, an optimum pH needs to be maintained, optimum temperature needs to be maintained, the raw materials need to be provided, you know, all of the medium needs to be created at that particular temperature. Only then that the DNA will be able to express that protein. So the bioreactor does that job. Okay, that is what the whole point of a bioreactor is. So of course, optimum conditions are provided for the production. Okay, so for example, if you want to isolate or express, want that bacteria, the rDNA, to express a particular protein, let's say an enzyme, of course. So of course, the rDNA would need all those specific conditions fulfilled to be able to produce the product. Okay, just simply having the rDNA there, it won't do anything, right? So yes, of course, you can see from here this steam for sterilization, this culture broth in it, there's an impeller which, you know, moves around. You need to keep sterile air, shouldn't be contaminated. There's acid base for pH control, so on. So all these requirements need to be fulfilled. All right, so a bioreactor does that. So yeah, a question was for growing microbes on a large scale. For a large scale production, yes, but that's the whole point, right? Eventually, you want to produce it at a bigger scale. Yeah, so bioreactor does the job. All right, let's move on. Question number 8. The correct order of steps in polymerase chain reaction PCR is, this appeared in NEET 2018. A. Extension, denaturation, annealing. B. Annealing, extension, denaturation. No, it's probably C or D. Yeah, sorry, it's D. This denaturation, annealing, and extension. Okay, so the answer is D. So what you see here is the other typical steps of PCR. Okay, so you have denaturation first. So the double-stranded DNA is split up. Then you have annealing of the primers. You have annealing of the primers over here. You notice these primers are attached in anti-parallel fashion. So if it's 3 dash over here, it will be 5 dash over here. Okay, against 5 dash over here, sorry, if it's 3 dash over here, it will be 5 dash over here. All right, so obviously you know the DNA are anti-parallel, so primers have to be placed that way. Okay, then you have your Taq polymerase and deoxynucleotides mixed. Okay, and then only extension will take place. So the primer is there to start, but you need the polymerase enzyme and deoxynucleotides to extend that strand of DNA to finally form a double-stranded DNA. All right. So over here they said 30 cycles. 30 cycles, if you keep repeating this process over and over again, it's like creating copies. Okay, it's like a photocopy. You just keep creating and creating and creating more. Okay, and it will be amplified to almost 1 billion times. So just imagine. So if you want a particular desired fragment, you can use PCR to create more copies of it. Okay, you have a particular fragment and you want to use that, clone it in a vector and so on, then you can create many copies of it. Okay, but yeah, the correct, as per our question over here, the correct order of PCR is denaturation, annealing, and then extension. Okay, so we've answered the question correctly. Yeah, the option D is the right answer. Can move on. Question number 9. The process of separation and purification of express protein before marketing is called, this appeared in 2017, it's called A, downstream processing. The other options, bioprocessing, post-production processing, upstream processing, are incorrect. So after you have used a bioreactor to create your enzymes or whatever product protein that is there, you have to separate and purify it. Okay, and also after that add preservatives, undergo it for clinical drug trials, or whatever, so on, if it's a medicine or and all that, or undergo it under quality control processes, testing, to make sure that your product is pure and it is saleable. Okay, eventually you have to sell it out to the market, right? So you need to allow all these processes to take place to finally form a product that you can market. Okay, so the process of separation and purification of express protein, sorry, before marketing is called downstream processing. So the answer is A. All right, so I pretty much explained it already. Yeah, so after the bioreactor process is done, then you undergo separation and purification. That's called downstream processing. So let's move on. Question number 10. Stirred tank bioreactors have been designed for, this appeared in NEET 2016. A, purification of product. B, addition of preservatives to the product. C, availability of oxygen throughout the process. D, ensuring anaerobic conditions in the culture vessel. So stirred tank bioreactors have been designed for C, availability of oxygen throughout the process. The answer is C. Remember I showed you the diagram, okay, of that particular bioreactor. So there are blades, right? There are blades. They look like fan blades, and it is placed in this bioreactor, and there's a culture broth. Now when you add your microbes to it, okay, this is simply, it's called, from the name itself, stirred tank. Okay, so this keeps moving round and round and round, rotates around the axis to allow oxygen to be available. Okay, because those microbes need oxygen to grow, okay, and to eventually produce the protein, right? So this oxygen, for availability of oxygen, it is designed in such a way that it keeps rotating on that axis and then eventually allows for oxygen to be available. Okay, so stirred tank bioreactor is used for production of biological products from raw materials, of course. The stirred tank promotes availability of oxygen. That's the whole point of that particular bioreactor. All right. So you answer the question though. Yeah, availability of oxygen throughout the process. Question number 11. Which of the following is not a component of downstream processing? This appeared in NEET 2016. A. Separation. B. Purification. C. Preservation. D. Expression. Okay, so which is not a component of downstream processing? That is D. Expression is not a part of downstream processing. It happens before that. Only after that, then there is separation, purification, and preservation. So the answer is D. Yeah. Of course, expression is not there part of downstream processing. That obviously happens in the bioreactor. After that, you have the downstream processing part. Okay, so preservation is done after the purification part. This preservation, and then that all forms a part of downstream processing. And then you have to be able to market that product, right? So you have to make sure that your product is up to industrial standard. Okay, let's move on then. Question number 12. The Taq polymerase enzyme is obtained from, this appeared in NEET 2016, A. Bacillus subtilis, B. Pseudomonas putida, C. Thermus aquaticus. That's the right answer. We already discussed this. D. Theobacillus. That's incorrect. So the answer is C. Okay, Thermus aquaticus. Yep, this is it. There's nothing much to explain. The Taq polymerase is used in PCR, and it's a heat stable enzyme. All right, let's move on. Okay, so question number 13. An analysis of chromosomal DNA using the southern hybridization technique does not use, and this appeared in 2014, does not use A. electrophoresis, B. blotting, C. autoradiography, and D. PCR. So if you're using southern hybridization, you does not include PCR. Okay, it will include electrophoresis, blotting, and autoradiography, but not PCR. Okay, so the answer is D. This is a good question actually. So southern hybridization, I think I already discussed this. It's when you use DNA fragments. So you run a gel. Okay, after it has been treated with restriction endonuclease, you run a gel, electrophoretic gel, and you will get your DNA fragments separated, right? Now, there is an instrument that is used by which if you use a nitrocellulose paper, a membrane, NC membrane, okay, you can transfer this into this nitrocellulose membrane. Okay, now once that is done, you place this nitrocellulose membrane in a liquid, a solution, okay, and put probes in it. Okay, that's autoradiography. Okay, they are radioactively labeled. Okay, they're radioactively labeled, and they will stick to the corresponding DNA fragments. So when you place this under an X-ray film, then you can see where those probes have been attached to, okay, which particular DNA fragment. So then you can identify. Okay, that is basically southern hybridization, but PCR is not included in this. If you have to answer the question, yes, PCR is not included. PCR is used when you want to amplify a particular desired fragment, okay, and which we are not doing over here, okay, when we are particularly talking about southern hybridization. So the steps basically include electrophoresis, then there's blotting, the southern blotting, followed by that there's autoradiography. Okay, so that is what is included in southern hybridization. Yeah, this is a good question though. The smiley face. Okay, well, let's move on then. Question number 14. In vitro clonal propagation in plants is characterized by, and this appeared in 2014, A. PCR and RAPD, B. Northern blotting, C. electrophoresis and HPLC, D. microscopy. So you're talking about in vitro, that's in lab, clonal propagation in plants. Okay, so clonal propagation, production of clones, is characterized by A. PCR and RAPD. Okay, answer is A. Okay, so PCR, we all know what PCR is, right? So RAPD is randomly amplified polymorphic DNA. It is a type of PCR. Let's learn it this way. Okay, it's a type of PCR, and they are both used for clonal propagation in plants, and they assist in amplification of genes which help in clonal propagation. Okay, and RAPD is a type of PCR where when we studied PCR, we add primers, right, to it. Now, primers are very specific in the case of PCR, but in the case of RAPD, those primers are not specific. That's the difference between the two. Okay, just think of it as this way: they're randomly amplified. Okay, polymorphic DNA, and PCR is very specific. The primers are specific to that. That's the reason. And of course, both of them are used to amplify that gene that you want, okay, to produce clones of it. So, yep, to answer the question, yes. Think of it this way also: northern blotting, electrophoresis, HPLC. HPLC is chromatography, microscopy. They don't help in clonal propagation. So, PCR and RAPD are the appropriate answers. This is also another good question. Okay, it's application based type of question. Okay, let's move on then. Question number 15. Which of the following is not correctly matched for the organism and its cell wall degrading enzyme? This appeared in NEET 2013. A. Algae methylase, B. Fungi chitinase, C. Bacteria lysozyme, and D. Plant cell cellulase. So, not correctly matched is A. Algae and methylase. So, answer is A. So now in many classes of algae, cellulose is the cell wall component. So you will ideally use cellulase. Now there are many classes of algae, so they have different components of cell wall. There's some classes that contain manganese, xylanes, alginic acid, sulfonated polysaccharides, silicon, calcium, so on. Okay, but methylase definitely doesn't fit into any of that. All right, so that is the correct answer for this particular question, because it's the incorrect match. All right. Okay, let's move on then. Question number 16. During the process, one second. During the process of isolation of DNA, chilled ethanol is added to. Okay, this was a repeat. Karnataka NEET 2013. We just discussed this, right? A. Precipitate DNA. Okay, so chilled ethanol is used to precipitate DNA. Break open the cell to release DNA? No. Facilitate action of restriction enzymes? That's incorrect. Remove proteins such as histones? That's incorrect. So the answer is A. Alright, chilled ethanol is used to precipitate DNA. Let's move on. Alright, so question number 17. PCR and restriction fragment length polymorphism, also known as RFLP, are the methods for, and this appeared in 2012, A. Study of enzymes, B. Genetic transformation, C. DNA sequencing, D. Genetic fingerprinting. Okay, so PCR and RFLP are methods for D. Genetic fingerprinting. Okay, so the answer is D. Okay, so PCR we know is amplification of that particular gene. Okay, and that can be used in genetic fingerprints. So genetic fingerprinting, also known as DNA fingerprinting, or DNA profiling, that's another term, is basically the analysis of that particular DNA. Okay, so we are analyzing that DNA, and they are very useful in determining disease, okay, causing genes. Okay, especially when there's a single gene that causes a disease, we use PCR and RFLP. Now, RFLP is restriction fragment length polymorphism is when you have this whole DNA fragment, all right, and they encode for the same. There are various DNA sequences over here that encode for that same restriction nucleus, or that's where the restriction nucleus endonuclease cuts. Okay, so if you have whatever GAA TTC, and you have your palindromic sequences over here, and let's say you have GAA TTC, okay, and you have a palindromic sequence here. So eco R1 will cut here, eco R1 will cut here, eco R1 will cut here. What will happen? So it will break down into 4 fragments. Yeah. It will cut here. Yeah, so you'll get various fragments over here. Now they are obviously polymorphs, right? They are coming from the same, they are all being cut by the same particular restriction endonuclease, so they are known as polymorphs. That's why it's known as restriction fragment length polymorphism. Okay, that's why it's called RFLP. Okay, and PCR, both PCR, obviously we know that it is the amplification of a particular desired fragment. Okay, so now these two particular methods are used for genetic fingerprinting. Okay, so now you can particularly analyze whether that that particular piece of fragment, where it is, it goes on and on. It's not like just PCR and RFLP are only one of the or some of the methods. Then after that you need to use southern blotting, and then you know what southern hybridization is, right? Now if, for example, you've done southern blotting, and then you can particularly find the gene that's causing that disease, okay, by adding probes to it. Okay, you add probes to it, and then now you can find that gene is deformed. Okay, the particular gene is deformed. Let's say you have a fragment length over here, and you this is a disease causing fragment. Okay, if it is a normal particular gene, sorry, then there will be no problem, right? The restriction endonuclease will cut as it is. Okay, if the restriction endonuclease cuts, then fine, you will get the particular fragments that are there. But if it is a diseased one, that means the DNA has been altered here. Yeah, the gene has been altered here. That means the restriction endonuclease will not cut there. That means it will not form a break there. Then you can see the difference between the two, between the normal and the disease gene. So you know that that particular gene is the issue there in that particular person. Okay, you can compare a normal person to the one that has a disease causing gene. So this particular PCR and RFLP are very useful in determining that. Okay, and then obviously you use southern blotting. So just PCR and RFLP don't make sense, but if you are comparing a normal to a disease causing gene, then you can compare the two. Okay, but to answer our question, genetic fingerprinting as in it is used for genetic fingerprinting, PCR and RFLP methods. Okay, let's move on. Question number 18. Which one is a true statement regarding DNA polymerase used in PCR? This appeared in 2012. A, it is used to ligate introduced DNA in recipient cells. C, it serves a selectable marker. C, it is isolated from a virus. D, it remains active at high temperature. The DNA polymerase used in PCR, it remains active at high temperature. That's the quality of Taq polymerase. Answer is D. It's a DNA polymer that is active at high temperature. All right, let's move on. Question number 19. The figure below shows 3 steps A, B, C of PCR. Select the option giving correct identification together with what it represents. Okay, so you have you have A, B, and C here. Option A, B, denaturation at a temperature. No, that's incorrect. Option B, A is denaturation at a temperature of about 50. No, it actually is 98. It's above 90 degrees. C, extension in the presence of heat stable DNA polymerase. Yes, that is correct. Option D, A, annealing with two sets of primers. No, that's incorrect. So option C is the right answer. Okay, so here you have that the PCR steps. Okay, so C is extension. Yeah, so you have DNA polymerase plus deoxynucleotides extension after addition of tag polymerase and deoxynucleotides to form the corresponding strand. Okay, so yeah, this is it. Denaturation, annealing, and extension. Okay, I guess I've answered the question. Yeah. Okay, let's move on. Question number 20. Stirred tank bioreactors have been designed for, this appeared in 2010. A. Addition of preservatives to the product. B. Purification of the product. C. Ensuring anaerobic condition in the culture vessel. D. Availability of oxygen throughout the process. So we already discussed this. It's a repeat of the previous question. The answer is D. Okay, so availability of oxygen throughout the process. Okay, so you have the bioreactors, you have the stir tank, you have the blades here that keep rotating on the axis, and so that can provide more oxygen. Okay, all right. I guess I've answered this question. Yep. Okay, so we've come to the end of this video lecture, and I hope you've enjoyed today's video lecture, and I hope you've learned something new today. And don't forget to like, share, and subscribe to MG NEET Biology. And I'm available on Facebook, Twitter, and Instagram, so you can follow me there for more updates. And I'll see you in the next video lecture. All right, bye.